Hello, welcome to part 3 of your lecture on the structure and functions of cells in the nervous system. In part 1, we looked at basic neural system organization, differentiating the types of cells in the central versus peripheral nervous system, also covering learning objectives 2.2 to 2.5. In part 2, we introduced the concept of the action potential and forces that are involved in its manifestation and propagation, covering learning objectives 2.6 to 2.10. In this lecture, we're going to focus on neurotransmitters, the chemical method of communication between neurons, contrasting the different receptor types, EPSPs and ISPs, which will be defined shortly, and also talking about autoreceptors and postsynaptic receptors, essentially thinking about the various types of communication. So in particular, we will be focusing on the structure of synapses, how neurotransmitters are released and where, their activation through receptors, postsynaptic potentials, the termination of postsynaptic potentials, the effects of these in terms of the neural integration in the organism, and also consider autoreceptors, axonic synapses, and non-synaptic chemical communication. So at the outset, some important concepts for you to grasp include the idea of the synapse, which we've talked about before. It's that junction between the presynaptic and the postsynaptic neuron in a chain of communication, with the synapse being the central point where they meet, right? There are different kinds of synapses. Some are electrical and some are chemical. And in class, we'll talk about their characteristic appearance, largely related to their spacing, right? Postsynaptic potentials are basically the brief depolarization or hyperpolarization, excuse me. So either depolarization or hyperpolarization that's brief. Neurotransmitters exert their effects by attaching to binding sites in much in a fashion very similar to a lock and key, not the kinds of digital locks and keys that we use today, but a physical lock and a physical key that fit like hand in glove. The ligand is essentially the term that's used to describe the key in this lock and key mechanism. It can be a steroid hormone or a neurotransmitter. This illustration from your textbook shows the neurotransmitter molecules represented as keys that fit into specific locks. So these keys are excreted by the terminal button, essentially, from a presynaptic cell, and they float around and diffuse and eventually bind to receptors. They're designated locks in that postsynaptic cell's membrane. So here you see that synapse sort of blown up and illustrated in figure 2.24. And you can see that this synapse is actually a terminal button from a presynaptic neuron connecting with the dendrite of the postsynaptic neuron. So this is an axodendritic synapse because it incorporates an axon and a dendrite. By contrast, we also have synapses that can occur between the terminal button and a dendritic spine. And so it's important to note that axodendritic synapses can take both forms. Also, it's possible to have a presynaptic terminal button and a postsynaptic terminal button, both attaching to the dendritic spine. So in this case, we have an exoaxonic synapse consisting of synapses between two terminal buttons. Neurotransmitters, it's important to note, are released into the synaptic cleft as vesicles, little packages of neurotransmitter that are made by the neuron, fuse with the cell membrane, and then spill their contents into the extracellular fluid. Here is a list of the common types of neurotransmitters, including adrenaline or epinephrine, noradrenaline or norepinephrine, dopamine, my favorite, serotonin, GABA, acetylcholine, glutamate, and endorphins. These are all water-soluble peptide neurotransmitters that require admission into the cell via some lock and key type mechanism. They should be distinguished against steroid hormones that because they're made of fat, have a cholesterol tail, can fuse at will with that fatty phospholipid bilayer that makes up a cell membrane and gain entry into a cell. Hence why steroids taken in extremely high concentration can be disadvantageous or harmful because they can gain access to the cell without permission. So here is another illustration showing you the details of a synapse. Again, you can see the microtubules along which the vesicles might travel from the cell body in the anterior grade direction to the terminal button of the axon. The little vesicles containing neurotransmitter eventually fuse with the cell membrane, release their contents into the synaptic cleft, which is that space between the presynaptic and the postsynaptic neuron, which is a little bit separate, allowing for the neurotransmitter to diffuse and eventually bind to its target receptors. And this illustration from your book shows two kinds of receptors that by which neurotransmitters gain entry into a cell. And the reason they want to get into a cell, and I'm anthropomorphizing neurotransmitters by saying they want entry, but that's how we change the functions of a cell, in part by changing the way that the DNA in that nucleus is transcribed and translated. How our genes are expressed is impacted by these neurotransmitters. So there's the ligand-gated ion channel, which you see illustrated on the left, and the G protein coupled receptors, which are illustrated on the right. So neurotransmitter molecules diffuse across the synaptic cleft, attached to binding sites called postsynaptic receptors. And once binding occurs, the postsynaptic receptors open neurotransmitter-dependent ion channels, allowing the movement of these ions can change the local membrane potential. And this is an important piece because not only can neurotransmitters affect gene expression, they can affect the action potential, the depolarization or hyperpolarization part of the neuron's electrical communication. So in this illustration, you can see how a binding site is situated across that phospholipid bilayer that makes up the neuron's membrane. You can see some ions, these little blue spheres, and a molecule, a neurotransmitter that is attached to its binding site, one little black circle. And you can see that by attaching to its binding site, it is allowing for the entry of ions through an open ion channel. So this is an example of an ionotropic receptor. The ion channels on the membranes are cylindrical structures, as you can see, that open to the outside and the inside of the cell. On the end of the outside of the cell, a small curve-like structure can be seen, and that is the binding site. When there is no molecule at the binding site, the receptor, the ion channel is closed. When a neurotransmitter attaches to that binding site, the ion channel opens. And these ions, as you can see, are represented as tiny spheres that move in and out of the cell via this now open channel. Receptor activation can also take the form of metabotropic receptors, which are considered indirect receptors because they don't directly lead to the admission of an ion, but rather through a cascade of events allow a change in the flow of ions into or out of a cell. So you can see this illustration shows a neurotransmitter molecule, again in black, but this one's much bigger, binds to a metabotropic receptor that then triggers a G protein that then binds to another structure that then triggers a second messenger that then changes the way the ion channel is working, allowing neurotransmitters to enter the cell. So in this instance, the metabotropic receptor is an indirect mechanism by which neurotransmitters can impact the electrical signaling between neurons. And you can see that this metabotropic receptor is located close to the G protein. It activates an enzyme that stimulates the production of a chemical called the second messenger. Changes take longer to begin and last longer through this mechanism than you might expect through the ionotropic mechanism. The flow of events again includes a molecule binding to the metabotropic receptor from outside the cell, the metabotropic receptor flowing into the G protein that is inside the cell, the G protein connecting with an enzyme, and finally the enzyme flowing to a second messenger, and the second messenger to the ion channel where some ions are present on the inside of the cell and may consequently be booted out of the cell. So I want to remind you that your textbook publisher, Pearson, has some helpful simulations showing the activation of receptors and also the termination of postsynaptic potentials. We're not going to spend a whole lot of time on that, except to say that there are 4 types of neurotransmitter-dependent ion channels, and these don't differ from the types of ion channels that we have talked about in the context of the action potential and electrostatic forces. They are one and the same characters in the cast, right? So a postsynaptic potential is determined by the characteristics of postsynaptic receptors and the type of ion channel they open, right? And so that's the important piece that I want you to remember And here you can see in an illustration that ions move during postsynaptic potentials. A molecule of neurotransmitter, again, illustrated as a black sphere, attaches to the binding site of all the ion channels. The outside of the cell is positive and the inside is negative. The movement of ions during the postsynaptic potentials goes, in the case of item A, a sodium ion, you see enters the ion channel. There's an influx of sodium that causes a depolarization or an excitatory postsynaptic potential. And in the case of B, you see a potassium ion leaving the cell, and the efflux, so influx is entry, efflux is exiting. The efflux of this potassium ion causes hyperpolarization or an inhibitory postsynaptic potential, an IPSP. A chloride ion in item C enters the ion channel, causes hyperpolarization, an IPSP. In item D, you see a calcium ion is entering the cell subsequent to the ionotropic receptor binding of a neurotransmitter. And that entry of the calcium ion, the influx of calcium activates an enzyme and, culminates in certain enzyme effects in this particular instance. So we've just defined ionotropic and metabotropic receptors and talked about the types of postsynaptic potentials. So an excitatory postsynaptic potential, an EPSP, involves the sodium channel being an important source of depolarization. And an IPSP, and I'm distracted because my potential, that word under inhibitory postsynaptic potential, has been partially erased by the illustration, but that IPSP stands for inhibitory postsynaptic potential, and it is involved in hyperpolarization. And you can see that ionotropic and metabotropic receptors can both have an effect on EPSP and IPSP. There is a little bit of a time lag in terms of the IPSP impact through the metabotropic receptor, but they both influence depolarization and hyperpolarization. So reuptake is what we call the rapid removal of neurotransmitter from the synaptic cleft by the terminal button. And there's also enzymatic deactivation, which has to do with enzymes that destroy or break down molecules of the neurotransmitter. And acetylcholinesterase, anything that ends in an ACE, is an enzyme. Acetylcholinesterase is an example of an enzyme that deactivates acetylcholine, the neurotransmitter acetylcholine. And here in this illustration, you can see again two cell membranes, the presynaptic cell and the postsynaptic cell. And at the top is the presynaptic neuron, at the bottom is the postsynaptic neuron. The presynaptic membrane at the top has a cylindrically shaped transporter on it, And following this transporter, the membrane makes a concave upward, right? There's that little wave. And this is marked as an omega figure. And it's the remnants of a synaptic vesicle that has released its neurotransmitter. So by looking at this omega figure in the surface of the presynaptic cell, we can tell that it recently released its synaptic vesicle contents and has spilled neurotransmitter out into the cleft. The space between the two membranes, again, is a synaptic cleft. The neurotransmitters present in this cleft are represented in tiny dots, and they exit through the transporter at the presynaptic membrane to the terminal button. So neurotransmitters in the synaptic cleft also are bound to that postsynaptic receptor on the ion channel at the postsynaptic membrane, and that's what we can see here. So now we are going to think about neural integration, which involves an interaction of effects, an interaction between EPSPs and IPSPs. So neural integration has to do with the rate of neuronal firing as it's controlled by excitatory and inhibitory inputs to the dendrites and soma. If activity of the excitatory synapse goes up, increases, the rate of firing will increase. If activity of the inhibitory synapses go up, the rate of firing will decrease. So you'll remember early on in this chapter, we talked about the withdrawal reflex and about inhibition of that withdrawal reflex under a circumstance where dropping a hot object can be more catastrophic, right? So this is essentially what's happening with neural integration. In this illustration of the let's say the soma we're looking at is a postsynaptic neurons cell body in orange, and lots of presynaptic axons touching on or synapsing with that neuron. You can see that there are excitatory synapses noted in red, right? And they're excitatory in terms of the impact of the presynaptic cell on that postsynaptic membrane. And the illustration also shows, so the excitatory synapses are represented as arrows that act on the dendrites and the soma, and they are therefore labeled activity of excitatory synapses produces EPSP, so excitation, excitatory postsynaptic potential in red, and you can see that the signal reaches the axon hillock, right, which is the source of the action potential. And in this illustration, we have the same neuron, the same postsynaptic cell. On the one hand, we talked about it receiving excitatory synapses. On the other hand, now we're looking at some inhibitory synapses. So on the blue side, cooling down the mechanisms, right? We see presynaptic neurons projecting onto this soma and resulting in an activity that's inhibitory, producing an IPSP. The neuron is sort of cooling its activity, turning blue, which doesn't really happen, but for the purposes of illustrating the point. And as you can see, the IPSPs have the effect of counteracting the EPSPs also experienced by that neuron, resulting in the inhibition of its firing. So in the case of autoreceptors, these are so generally speaking, right, a neuron will secrete neurotransmitter. The vesicle will release the neurotransmitter into the synaptic cleft. It will be bound by the postsynaptic neurons. Neurons have the potential, thankfully, to also bind their own neurotransmitters that they release. And this is kind of like, hearing your voice when you talk. If you are speaking in a microphone and the feedback you're getting is of some distorted audio, then you will adjust your voice, maybe pull your mouth away from the microphone, and so on and so forth. So these autoreceptors kind of function in a similar fashion. They enable the neuron and its effects to be influenced by the neurotransmitters that it releases, in as much as it's influenced by neurotransmitters that are released by other neurons, with which it forms a synapse. This has the effect, autoreceptors have the effect of regulating internal processes like the synthesis and release of neurotransmitters that are inhibitory. In the case of the axo-axonic synapse that we mentioned earlier on in this lecture, you can see that the terminal button of axon A, right, the presynaptic axon A, can increase or decrease the amount of neurotransmitter released by the terminal button of axon B from presynaptic neuron B. So it gets a little bit fancy, but the important thing to know is that axo-axonic synapses change the amount of neurotransmitter that's released by terminal buttons in a manner similar to what the autoreceptors are doing. And therefore, they can produce presynaptic inhibition or presynaptic facilitation, enhancing or inhibiting the effects of surrounding synapses. I have this table included in the lecture to show you, generally speaking, that the chemicals that are involved in chemical communication between synapses fall into two. So I talked about peptide or peptide hormones and neurotransmitters and steroid hormones. Those are two different categories. Any chemical, when it's in the brain, can be referred to as a neurotransmitter, provided it's exerting functions through the neuron. So steroid hormones are engaged in neurotransmission, as well as these peptide hormones. But in keeping with our discussion of the excitatory or inhibitory effects of neurotransmitters, it's important to know that they fall into, these different categories of being neurotransmitters or neuromodulators. So examples of neurotransmitters included in this table are the amino acids, glutamate or gamma aminobutyric acid, GABA. Both of them are produced in the brain. GABA is synthesized from glutamate by glutamic acid decarboxylase, TMI. The important thing to note is that that is categorized as a neurotransmitter. By contrast, there are certain neuromodulators that are also chemicals produced by neurons. So in the case of noradrenaline, which is one of the catecholamines listed here, it's produced in the locus coeruleus, a part of the brain. But the difference between neuromodulators is that their effects tend to be a little bit more dispersed, a little bit more long-lasting, and less and more persistent, I guess is the word I'm looking for. So neuromodulators are chemicals that are released by a neuron. They travel farther, disperse wider than is the case with more neurotransmitters, So based on this definition, steroid hormones would be categorized as a neuromodulator, right? Because they have, they're just secreted, for instance, by the gonads, travel through the bloodstream, get into the brain as they wish, affect lasting change in neural functioning. So I talked to you a little bit about neuromodulators. So hormones are, again, any chemical, there's a broad definition, any chemical that influences actions of the cell, affecting the functions of a cell, gene expression within a cell, growth and sort of concerted, coordinated functioning of the tissue. So you can see here that there are a number of different kinds of hormones that are produced even in fat tissue that regulate varied functions of the body, including growth and immunity and reproduction, as well as behavioral function. So hormones generally are secreted by endocrine glands or cells found in other parts of the body, right? So when a chemical is produced in the brain, we refer to it as a neurotransmitter, a neurochemical, a neuromodulator. And in the case of hormones, traditional hormones usually have a receptor, right? A receptor that is specific to them, much like the lock and key mechanism that's described before.